那今天的专题演讲跟这个礼拜的专题演讲有一点点不太一样哦。这礼拜专题演讲是由这个伍焜玉院士学术讲座遴选委员会所规划的。这个讲座是我们大概在2020年的时候由江安世老师、江安世院士所发起。那我们先请江老师帮我们简单介绍一下这个讲座的起源。大家好，很幸在这边每一年都有机会来介绍会议给大家认识。刚才那个陈振辉提说，其实做研究就是讲故事，发掘生命的故事。那一个role model， 我的 role model 是伍焜玉。那伍焜玉的故事，他在台大医学院毕业医学士，然后在耶鲁硕士，去英国伦敦大学博士。这之后呢，他在美国大约30年左右行医，然后当医院或学校的主任中心主任所长等等，然后回到台湾94年当选中研院士。在台湾有各式各样的工作国卫院的院长很久的时间。他的主要的奇幻之旅就是其实他很年轻的时候就发明了这个血小板凝结的这种计算的方法当时候发在 Lancet， 这个是台湾那时候时尚科学还很刚开始的时候那在国际上面被誉为是血小板先生，得到国际的这种重视，甚至好像德州还有一个伍焜玉这个日专门纪念他这个发现。那我今天要讲的是他2015年他得了台湾最高的科学的荣誉，就是总统科学奖。很喜欢做点事情。那时候我就想了这个法子在台湾有很多故事但是好像还没有一个年轻人的研究的故事是从下而上，所以才有了这个伍焜玉的学术讲座。说如果我们找几个年轻的教授我们这些正教授都不要几个以外都不要几个年轻的教授，然后让他们继续挑他们最想要学的台湾的年轻的最杰出的科学家的这个人选是谁今年已经第3届了，很显然我们每一届挑到的都是很独特的人陈振辉会有其他人来介绍跟大家介绍这个宗旨。跟伍焜玉提这个 idea， 伍焜玉喜欢极了，就把他的总统奖所有的奖金都捐出来了就放在我们生科院伍焜玉特别的，他是 MD PhD 他特别的觉得说医学或者要好，一定基础科学要好。所以他特别的希望说用这个方法来激励对医学基础医学有兴趣的这个学生所以我们在挑选的就是说，而且还有就是说最好是这个有某些技术这个又特别的好，因为很多陈诺贝尔奖给他看是因为这个技术的创新，当然没有一定因为最后还是看看当时候这些年轻的最优秀的学者就是我们刚回来的这些教授，他们最想要而且他们最崇拜的这个年轻的在台湾的偶像是谁所以以上这些就得到伍焜玉的这个认同每一次他还为这个事情专程的有一次大概 get together， 希望把这样的故事一直在清华这个地方一直的延续下去，也希望有更多的学生能够享受做研究的过程。这个 program 除了早上的演讲，还设计了下午可以跟speaker 有很多的互动这个 program 执行内容就交给主持人继续往前介绍到这里，谢谢。
谢谢张老师好，那现在接下来就让我来介绍今天的讲者陈振辉老师It's my honor to introduce today's speaker, Dr. Zheng Hui Chen. Dr. Chen got his bachelor degree and the master degree from National Yang University. Then his PhD degree from Dartmouth College. After his postdoc in Duke University, he went back to Taiwan and joined academic syndicat in 2016. And since then, he has already received a lot of awards, including Young Investigator Research Award from MOST and the Career Development Award from Academia Syndicat. And this year, he just received the Outstanding Research Award from NSTC, and also he is being selected as an ENBO Global Investigator. Okay, that's a lot of accomplishment. Okay. Dr. Chen's research is mainly focused on the development and the regeneration biology. They developed a new cell-taking, multicolor cell-taking system, which allows them to tracking cell behavior on the whole animal scale. So by using this technique, they recently discovered a new form of the cell division, which was published in the Nature last year. So I personally believe that this finding will be the next version of chimera biology. Okay, so I would like Dr. Chen to tell you more detail about his research, but before that, let's invite our dean to present this award to Dr. Chen. 请院长给我们颁奖。Okay, it's about the story time. So please join me to welcome Dr. Chen for his talk.
I feel very honored to be here to tell you what we have been doing academic syndicat. And I would also like to take this opportunity to thank伍院士、江院士、院长和林学委员玉俊，还有遴选委员会的背后这些成员哈。They really spent a lot of time on making this, preparing this lecture, and I really appreciate the recognition. And today, I wanted to tell you something about fantastic fish and whale planet. So I have two specific goals for the day. First is I will highlight the total approach we established in the lab, and also its combination with the zebra fish model. And the second goal is that I will introduce you two new cellular mechanisms that we identify through the total approach for supporting skin and muscle growth in zebrafish. In the beginning, I want to make clear of the terms that I will use in this presentation. So firstly, for people who study cell behavior, we use in vitro and in vivo to refer to studies of cells that are either outside or in a living organism. And when people talk about in total, right, sometimes the definition, right, is a bit loose. But here, specifically, right, I use in total to refer to studies of every single cell in a living animal. And later, I'm going to tell you why we consider the in total approach is very important. Okay. And as a developmental biologist, I use a microscope a lot to study cell behavior. So every time when I look at how the individual cell behave under the microscope, right, a very small field of view, I often wonder what I study, what I look at, are they representative? Or maybe oftentimes I'm just like a blind man facing an elephant. Only see pieces of the information, and sometimes even misinformation, but not the real picture, the true biology. So since I set up my own lab at Academic Sinica, I keep thinking about how I can be less biased to study cell behavior. So here I want to highlight that the zebrafish model provides a very unique opportunity. The size of the zebrafish at larva stage is about 3 to 4 millimeters in length. Even all the way to adult stage is about 3 centimeters. So the idea is that if I can be creative in terms of cell labeling tools, in terms of imaging platform, can I use a zebrafish as a gateway to study all of the cells in a living animal as development and regeneration occurs? So that's the whole idea. So to increase the number of the cells we can monitor simultaneously in a living animal. Over the past several years, my lab very much focused on the brainfold technology. And this tool is not new, first developed by Joanne Levy when he was in Jared Richmond's lab in Harvard in 2007. And basic idea is this is a reporter cassette flanked by 3 fluorescent proteins, flanked by 2 different pairs of last P sites. After trigger genetic recombination by recombinase, the cassette will either express the red color, blue color, or the green color. And the beauty of the system is that if you are able to put in multiple copies of the cassette into your system, let's say 3 copies, you can generate about 10 different colors to label your cell type of interest. And the thing we try to do in the lab, right, is try to push the limit of the system. Try to squeeze in as many copies as possible, let's say 20, 30, or 40 copies. The idea is that theoretically we can generate about more than 100 different colors to label a homogeneous cell population. And that's the things we are trying to do in the past several years. And here I want to show you how we apply the idea firstly in skin cells. And in the lab, we create a transgenic animal we call the pumpkin. And this is a stable transgenic fish. I'm happy to share the fish to any one of you. You grab the fish and bring it to a lab, you'll get the same results we have. Okay. And in this fish, we can generate about 70 different colors to label the entire skin population. And by the way, the project is spearheaded by a very talented graduate student at the time, Kei in the lab. Now she already moved on to her postdoc in Cambridge. So using this transgenic animal allowed us, right, to not just to monitor one single cell, but the entire skin population in a living animal. So after trigger labeling, we can precisely determine how many times a single cell has divided. And we can determine their division orientations. And also because these images were all captured from a confocal microscope, so we also can quantify three-dimensional information, such as changes in cell volume and changes in cell height. And we are not only doing that on a handful of cells, we are doing this on the whole animal. So we can know that all these cellular dynamic features, are they differentially regulated on different body regions or not? Okay. So from this extensive data set, we accidentally found that there's a subset of skin cells. We call it SEC, superficial epithelial cells, are able to divide twice to produce proteins without the DNA replication. And let me play the video for you. So in this case, he was able to repeatedly sedate the same animal, track the very same cell over about a 10-day period, and to catch such unique kind of division. And she found that these superficial epithelial cells will either divide once or divide twice before they shed from the body surface. Okay. And because this cell population has been shown to be post-mitotic, to have no DNA replication, but you must think about in our scenario, right? If you see this cell divide and this cell has been reported with no DNA replication activity, would you think that maybe something wrong with your system, or you won't suspect that there will be a new type of cell division, right? So that's the idea we have in mind. So we really want to validate this by ourselves. So to test, to confirm these populations really have no DNA replication activity, we create two additional transgenici, one specific labeled stem cell population, the other one labeled superficial population, and we do the in total imaging on the whole animal. Okay. So by doing the simple, very simple EDU assays, we found that the stem cell population, you can always detect about 1 to 5% of the positive EDU positive cells. However, if you look at the superficial epithelial cell population, we fail to identify from total about 20,000 cells. We fail to identify not even one EDU positive cells. Okay. So based on this data, we can start to make some very bizarre predictions. So if the cell division can occur, indeed can occur in somatic cells without DNA replication, the very simple prediction would be these cells must have a reduced genomic content. So here, he did a very simple dating assay, just stand for the genomic DNA. And what she found is that indeed, if you compare divided versus non-divided cell, there is about 60% reduction in terms of the genomic DNA content. And she also found that in addition to reduction in genomic DNA content, these cells also have less histone is labeled, and also has reduced the apical surface, reduced cell height, and reduced the cell volume. So based on this collected evidence, we conclude that this division is neither mitosis nor meiosis. And that gives us a very unique opportunity to name this type of division. And later, after several months of discussion and thinking, we decided to call this new type of cell division asynthetic division. We use asynthetic to highlight there is no DNA synthesis during this division process. And we should use fission simply means we just have no idea how this division is possible. Because the cell division supposedly should be a tightly controlled process. Like how could a cell are able to divide without the replicating DNA? What's going on with all these cell cycle checkpoints. Why all of a sudden all these rules that we know about, it doesn't apply to this cell division mode? And the second prediction we made here is because one single cell can divide twice to produce 4 cells. So as a diploid cell, at some point, chromosome cannot be aligned properly during the division. So we predict that there might be a detectable chromosomal segregation defect. And on the left panel here, so this figure I'm going to show you time-lapse images. And on the left panel here would be the time-lapse images we capture from a typical basal stem cell population. So you can see that the cell division see as normal, chromosome will condense and then has a very clean separation. However, if you look at the FEC, at the time we were so surprised to see that you can detect all kinds of chromosomal segregation defects that normally you will only see in cancer cells. But now you are seeing this in skin cells, in developing zebrafish larvae during normal development. And again, using the line vision approach, we were able to detect anaphase bridges, lagging chromosomes, micronuclei, and tripolar segregation during this when this type of division occurs. Okay. And in order to quantify the ratio of this, how many, in order to quantify the occurrence of the segregation defect, here can further conduct the alpha and gamma tubulin staining, try to categorize the mitotic spindle and to put a precise numbers on the frequency of the abnormal division. And what she finds that at any given time, she can detect about 20% of the cells that will have, at the metaphase, will have the tripolar spindles. And the next question that we have is think about that over the past 180 years, only 2 tidal cell division have been discovered. But we are the group identified a certain type. So we start to, we also don't quite believe our data. We want to know, right, is this a division over the place, or it only occurs during a specific developmental time period. So for that, further develop a flow cytometric-based assays. In this case, she do the cell sorting, identify the EGFP positive SEC cell, and specifically focus on the light cells. And later, because we know these cells will have a reduced genomic DNA content, so we can focus on the sub-2N population. And here we also need to check the 4N population as a control. If these cells, they divide, and because they have DNA replication, then we should be able to detect the signal here. And we try to check this cell population about the ratio across different developmental stages. And what we find is that, no surprise, if you look at the 4N population, the real is always very low, close to the detection limit, suggests that these cells are indeed or are post-mitotic, no DNA replication activity. And yet, when you look at the sub-2N population, we found that this population is very low during early development and is very low at adult stage. Only peak at a very specific time point. In this case, right, day 14 post-fertilization. Okay. So the next question, okay, so good to point out that by day 14, right, we can see about 50% of the cell undergo a synthetic fusion. So it's not a rare event. During a specific developmental time period, it can have up to 50% of the whole total population has undergo such a division. Okay. And but the next question we have is then what's special about day 14, right? why it's not day 8, why it's not day 21, but day 14. So I discussed with Kei and suggest maybe it's good to go back to look at the fish growth during this period of time. And what she found is that during this period of time, this period of time in zebra fishes considered a portion the only growth period. So you can see the animal grow rapidly if you check the trunk surface area. And so the fish grow rapidly day by day. And when you look further to specifically look at the growth rate, what you found is that the growth rate will peak at day 14, right at the day 14. So here the data suggests we see the most synthetic vision at day 14. And the animal also happen to grow at its fastest rate at day 14. So is it possible that the synthetic vision serve a physiological function? And to test this idea, we come out a model here by following a very simple scaling law. Somate a single cell, simply divide once without making any new materials. Just very simple scaling law. We assume the cell maintain the same shape during the division, but no synthesis of any new materials. Simply by doing so, the apical surface can have about 26% increase. And if a single cell can do this twice, like what we identified from our imaging data, the apical surface can increase about almost 60%. And very simple simulation. We did all of this actually, the first version all by hand. We found that this increase is not sensitive to changes in cell height or changes in cell shape. So when we plot the simulation data over the actual experimental data, what we found is that, wow, they match very well. They match very well. Okay. So in order to establish the causative connections, so we in the lab we further develop protocols trying to manipulate fish growth. Can we make the fish grow very fast, even faster in a short time, or can we slow down their growth? Can we really establish causative connections between animal growth and the synthetic vision? If our hypothesis is correct, our hypothesis is that synthetic vision is a way to quickly increase the body surface area to protect animals, to its skin integrity. Because the basal epithelial cell can undergo normal division to expand their surface. But for differentiated cell population, this choice is not there. So maybe that's the reason this cell population would do this kind of cell division. So later we figure out by manipulating fish growth density, either the high density, medium density, and low density, we can push fish to have the body length. If you measure the body length, has a 60% increase, or the body surface area have a 55% increase in just 2 days. Okay, this is the protocol we established in the lab. And by doing so, when you go back to check the synthetic vision, you can see that the coverage, the SEC coverage rate remain constant at 90%, and yet the cell number almost double. And the increase is all driven by a synthetic vision. So you can see there are more 3-cell and 4-cell clones being produced. So from all, and if you compare divided pool, compare the high density versus low density scenario, when fish grows slower, slowest and fastest, you can see there's 300% increase in terms of divided cell population. So from this data, we conclude that the surface growth can regulate SEC division. Okay, and from these findings, we want to dig further into the regulatory mechanism. Right. So what would be the connections between the body surface growth and the cell division? So we hypothesize that maybe during the rapid growth conditions, the body surface tension play a key trigger for this kind of division. So to address that, to test this idea, we further developed another transgenic line to specifically try to check the changes in body surface tension. And we use the live scarlet reporter to see, to monitor the cell cytoskeleton dynamics. And the bottom here, I'm going to show you how we measure the body surface tension using the recoil assay. So by using specific, very fine laser line to create wound on the fish body surface at different developmental stages, we can see that indeed during development, the animal body surface has a very dynamic change. Compared to day 8 versus day 14, you can see the body surface tension. If you look at the recoil velocity and the maximum recoil distance, both data suggest that the body surface tension are higher at day 14. So, and to further establish the molecular mechanisms that might connect the tension change with the cell division, we further conduct a small chemical screening and try to identify specific, we hypothesize try to identify specific molecular regulators that are important for the division. And from the screening, we found that 2 chemicals. One is the gadolinium and one is the iodine-1. One can either replace or enhance the division. And the gadolinium happens to be a stretch-activated ion channel inhibitor. And iodine-1 is an activator specific for the piezo-1, also one of the stretch-activated ion channel. And we found that the drug treatment can promote or replace exocytic fission without affecting animal growth. So this data suggests that the effect from these chemicals probably is more direct. It's not through a non-specific perturbation on the animal growth. And to further pinpoint whether there is a gene, an ion channel specifically involved in this process, we further developed the GAIA-mediated division method, knockdown method, to specifically remove the piezo-1 under the mosaic scenario. And we find that by doing so, so single injection, we can remove the piezo-1 down to 53%. And after that, we can see there's a reduction in exocytic fission. And because the knockdown is under the mosaic setting, so we also see no influence on the animal growth, suggests this perturbation, like the effect probably is more direct. It's not non-specific. Okay. So from all these findings, I would like to give you a quick sum up here before I move on to a short story now in currently under revision. And that is we create a transgenic tool, we call it the pumpkin. And that allowed us to achieve, for the very first time, the total monitoring of all the skin cells in the live animal throughout the entire post-embryonic growth period. And what we found is that we provide the evidence some small somatic cells can undergo cell division without the DNA replication. And we have a very unique chance to name this kind of cell division. We call it asynthetic fission. And we also found that a very simple scaling law can predict changes in cell size and changes in cell height and changes in apical surface. And we also provided that the tension and ion channels, specifically stretch-activated ion channel piezo-1, play a key role in regulating such division. Okay. And here, I would like to bring up a few questions to let people think about it before I move on to the second part. Should you care about a synthetic vision? So, up to now, there are still many things unknown about this type of division. We really have no idea whether the division is specific to skin cells or not. Or put it more broadly, we don't even know whether the division is specific only to liver fish. Okay. But if the answer is no, can we find a division? Is this in human and mouse? If so, can we speculate that the division is involved in some kind of human disease conditions? Right? So these are all questions, right, good to think about. And these are all future research directions, right? We are very excited to hopefully we can find good collaborators to move forward these directions. And also, I want to provide you some tips to spot a synthetic vision in your system. As I know that at Tsinghua, a lot of people also apply imaging approach to study cell behavior or to study the development. A lot of people use model systems here. So I want to provide you with 3 tips to identify synthetic vision. The first is because this division line can occur without a DNA replication. So by doing very simple DNA scan and carefully quantify the content, if you quantify enough numbers, you'll see whether you can see whether there will be a proportional reduction in genomic content or not. And also we found that the genomic content is tightly coupled to cell size. So good to pay extra attention to extra small cells in your system. Because believe it or not, I think there are a lot of literature already out there talking about if cells can undergo interpretation. Actually, cells will become bigger. Somehow, for some currently unknown mechanism, DNA content is highly coupled to cell volume. And that we also see that during synthetic vision. And the last one is this is a very error-prone process. So by doing alpha, gamma scanning or others, or simple the fixed tissue to look at the daily signal during the meta phase, actually you can see there will be a high occurrence of chromosomal segregation defect. Okay, so here I provide you 3 tips, just in case you are interested in looking into this kind of cell division in your system of interest. Okay, so the second part, just 3 or 4 slides, I want to highlight the in total approach. Right? If you are the first to do it under a light gene content, there are always expected things to be identical. So this is another project, another fish we create in the lab. We call it the pencil. We also give it a name, the pencil fish. And the project is spearheaded by another very persistent graduate student, Ubekuma, in the lab. So in the pencil fish, we use the fish to label all of the skeletal muscles in a live zebra fish. And I'm going to show you how this fish looks like, how this fish actually looks like under the microscope. So you can see that this is not a Photoshop image. So this is in the fish, the images that we capture under the confocal microscope. And you can see using this pencil fish, they was able to simultaneously monitor about 3,000 myofibers at the same time. And he can see how this muscle behave in number, in size, during both development and regeneration. Okay. And again, simply by doing imaging long-term tracking, they have some very unexpected findings. What he found is that regardless of the anatomic positions, all the muscles would get fully lava myofibers would get fully eliminated during a post-embryonic growth period. So here I'm showing you the heterophyte myofiber cell animal tracking. You can see there is a gradual loss of the myofibers. And this is the craniofacial myofibers. And of course, in order to convince reviewers, we also monitor both the eliminating myofiber and also the pre-existing the new myofiber. We also have the tools to monitor both populations at the same time. I just want to here give you a quick sum of our findings. So what we found in this study is we try to propose that there is a new type of muscle growth mechanism that nobody knows about it. That is, we propose that zebrafish as a vertebrate animal has two sets of muscles. One is larva myofiber, the other one is adult myofiber population. One is transient, the other is permanent. And there is a full replacement going on during post-embryonic period. And we call this, we give it an acronym, the SWAP. It's like, the muscle, we consider the muscle is one of the biggest tissues, right, a vertebrate animal has. And now we are seeing a clean swapping going on. And this process actually often reminded me about the human teeth development. Like we have baby teeth, we have adult teeth, and there is a changing process, right? It takes several years to complete. At any given time, if you look at your teeth, right? Look at the teeth in a child, actually you would not know there is such large scale replacement going on, because although it's on a large scale, but it happens in a very slow and steady manner. We want to say that actually the muscle growth in zebrafish occurs just like that. There is a slow and steady full replacement. To the adult stage, all your muscles all get a fully remake up. Okay. It is like an under skin metamorphosis process going on. And nobody knows about it until now. Okay. And this study is currently under revision. Yeah. Okay, so I hope I would have achieved my goal of the day. I highlight our total line vision approach and the zebrafish model. And due to time constraints, because in the lab, right, we think that this rainbow-based approach, right, is not so easy to use. And we, for every tissues and organs, right, students or people in the lab need to spend almost like 1 year to identify one good stable transgenic line for doing this. And we believe that probably in this entire world, right, nobody will be lab stupid. So we think that we can do it, only we will do it. So we want to do it on every single cell type and organs in zebrafish. And I can tell you, for every organ tissue we look at, there is always something infected actually we can identify. Okay. And today, I only share with you only about 2 of them. One is about the skin cell behavior, the other is about muscles. And I want to put out a question, especially to the student here. What can a cell do in vivo? Anyone want to help me do that? What kind of behavior, if you think about cell behavior, what kind of behavior can a cell display in vivo? Any students wanted to answer this? Oh, moving, okay. Yeah, migration is one. How about others? Oh, die, yeah. Apoptosis is one. Very good. How about other behavior? very good communication, more high layer communication, but there are many different kinds of communication. Okay, very good. So usually when we think about major cell behaviors, we think about proliferation, differentiation, migration, and cell death, apoptosis. But think about it, if you look into literature, usually cells as a population, they can collect, they can do many things. They can do hypertrophy, simply become bigger. They can have cell-cell fusion. They can have indole replication, dedifferentiation in terms of the cell identity changes, dedifferentiation, transdifferentiation, and sometimes even live cell intrusion will also happen. Not through apoptosis, simply this cell just be screwed out of the complex. And now we also know that they can undergo a synthetic fission and collectively swap. And I want to highlight that if you think about development over regeneration, actually underneath at the cellular level, only the combination of all of these, right, will make this biological process possible. Have you thought about all this in your model of interest, in your system, or you only focus on one or two of them? So good to think about it. Okay, there are still many things to be learned in the modern cell biology. Okay, and here I just another thing to put out here, and later to also echo Jiang Yuan's research direction. So this says this is our single cell, the length, the size of the length is about 30 microns. And many people are trying to push imaging resolution, try to look at as small as possible, and to look at the intra-organelle dynamics, mitochondrial dynamics. But these are all our interests. But I want to say that maybe going toward the other direction, try to look at as many as possible. Let's say like Jiang Yuan's case, right? Look at the whole brain, right? Like in our case, right? Look at all the cells in the live animal. Going big, right? Going big can also bring many have chance, right, to make many aesthetic to have many aesthetic findings. Okay, do you want to go big or go small? With that, I would like to thank the lab members for today's talk. I want to thank in particular Lei and Pei, and also our wonderful collaborators, both from Dr. Cherry, Center, and Peggy, and also funding support from ICOD, Academic Physica, and MSCC. With that, I will be very happy to take any questions you might have. Thank you.
Thanks for this amazing talk, and now we open for questions.
Please.
Thank you for your talk. I learned a lot. And so I'm interested in that the synthesis fission cell, does that have a normal function cell, or they just have a particular function in this cell?
I see. A very good question. So let me rephrase your question. So these cells, right, they don't have normal DNA. They really are functional cells, or maybe we just step on a new kind of cell death process. Right. Yeah. Okay. Very good question. And I must say that this question, why I say it's good, right? Because I got asked about this a lot. And the answer, actually, I don't really have a good answer for that. But I want you to propose something to think about. Like think about our red blood cells. Has no nuclei at all, continue functioning in our body for 120 days. Are they useless cells, or are they important cells? Like, good to think about that. And now, in the lab, we also have some research projects, right? Try to go look into lab a particular question. Right? For instance, in our data, we only have the modeling data support that since this is a very efficient way, a full goal, yet very efficient way to increase the body surface, right? But at the same time, the DNA will become a normal, and if the chromosomes of DNA become a normal, usually the typical size of a cancer cell, right? So why would skin cell risk to have a normal DNA to do such divisions, right? Is it because if they don't do that, then maybe the skin barrier function will become compromised during development? And now we do have a project in the lab, right, trying to look into that to assign more specific physiological functions to this type of division. Thank you.
So my question is whether there's organelles in these symmetric tissue cells, for example, like whether they have or mitochondria inside of these cells.
I see. So this is also again a good question, but we haven't looked into it. So now that we can see this divided cell after the divide, right? They still can stay on the tissue body surface, right, for about 3 weeks period, right, before they eventually shift on the body surface. And whether this is an actively regulated process or is a passive process, right? And whether the other internal organelles, right, within the cells, right, how will they distribute, right, during the division, right? That we all haven't looked into. Now how we done, right, is we already conduct both the single cell sequencing at the DNA level and also the bulk RNA sequencing, right? And then now we have some specific candidates that we wanted to test. It seems like there are some genes, right, things can actively regulate the division, but we will need this is all very preliminary. We will need more data to support that.
Thank you for the wonderful talk. I'm a cell biologist, and I think the one key question is whether this event is a zebrafish skin cell specific event or it could happen to other cases. So I think it's possible that there is a certain tension force threshold critical for a synthetic fission. So during the early development of zebrafish, the skin cell can kind of over this super high threshold and trigger a synthetic fission. So if this hypothesis is correct, it's probably you can use organoid or other kind of cell type and trigger such a super high tension force to see whether you can see such a fission under your control.
Yes, very good question. So actually we have a side project going on, try to address and let me rephrase your question so that people understand it. So the thing is, the vision question he said here is also important one. So that is this division, why this division would occur is because the fish growth somehow provide a very unique scenario, right? Tension very high on the body surface, so the cell can do this, right? And usually we don't see this type of divisions, right, during for other animal or in other cell types. And we now collaborate with the physics department at academic center. We try to establish in vitro stretching system to see that whether we can couple the tension changes, right, with the division, and we will do all these in vitro and try to see, right, whether we can get some more precise result to address that. Yeah, and at least good to think because I just 2 months ago I also was giving a talk in skin meeting, the international skin meeting, and talked about this, and we talked people ask about if you want to find such division, right, where in the skin or where in the human tissue, right, we should look at. And one thing good to think about is even our skin, right, are very different from zebrafish skin, because zebrafish skin does not have the keratinized dead cell layer. And our skin, right, we have the keratinized dead cell layer. But think about it, from very simple histology, people already know that the skin cell, right, when they differentiate from the bottom to the top layer, they will gradually lose their nuclei. And nobody really studied this process so closely. Is it possible that actually this process, right, somehow already this type of normal division is already part of the integrated process during a skin stratification. So there is something good to think about. And also, when we talk about skin, we think about our outside skin. But actually, reproductive skin, the outermost surface, right, is covered by mucus and the stratified structure, no dead cell layer. And the structure-wise, right, is very similar to our mucosal epithelium, like the epithelium inside of our mouth. And that, right, maybe is a good place to identify synthetic vision. Okay.
Thank you very much for this very interesting talk. And I think you're very lucky to find new cellular process. I have a couple of questions. One is, we all know that the cells, normal cells, when you grow it in a tissue culture plate, then they have the mechanism of contact inhibition. And so once they contact each other, become confluent, then you will stop division. And certainly when you produce a womb, then you will start to divide again. So I wonder whether this kind of in vitro model can be used to perhaps study whether in a mammalian cell, normal mammalian cells, you can actually produce this so-called asynthetic fission by perhaps applying tension or things like that. I think that's probably something you can do. Right? Yeah. Another question I have is that normally when we think of the cell division, we think of mitosis and meiosis. And so, for example, during the spermatogenesis, oogenesis, then you have that kind of process built into the system. So for a period of time, you do have cell division without chromosomal division, 4N, and you divide into 2N, and eventually into 1N. And so I'm just wondering, when you consider that as asynthetic fission,
Okay, so these are two questions. Let me answer one by one. So firstly, whether the cell culture would be a good model to study this. I think this is a very interesting things to think about, because the model you talked about, right, people culture the cell to conference and create and generate a square room, right, to let the cell as a vitro healing model. But oftentimes, people only use this model to look at the migration and stretching, right, and use it to, and sometimes proliferation. But people do not, right? They say if this kind of division does occur at low frequency, actually people won't know about it at all. Right? This is just protect only on a fraction of cells. Right? So I think there is opportunity, right? If people not know about this division, this, and go back to, I think for sure there will be already people looking into their system to look for such kind of division. Because we simply just don't know such division is. Any cell division you see, you just naturally assume it's a mitosis. Right? So I think this is just actually even such a simple basic process, right? It's understood. And the second question also is good. So you are talking about meiosis scenario. Right? Meiosis scenario, DNA will replicate once and then later divide twice. And during the second division, right, it's like a synthetic division. Right? This is also in the intriguing idea. And now we have some single cell DNA sequencing data. And that suggests that actually this division is very unique. We found that the chromosome is all randomly segregated. There is no defined patterns we can identify from 3,000 of cells within us. So that also put such division in a somehow very unique category.
So it can be considered as a defect, right?
A defect.
A defect.
A defect.
Segregation defect,
distribution, we found that if we now in our experiment, we have a specific probe to target specific chromosomes. So we found that the segregation has no any specific rules. So it's all random. It's all random.
Yeah. Another question is concerning the muscle.
Yes.
Because I used to look at xenopus muscle. Xenopus as a developmental biologist, it's a very good model for studying development.
Yes.
And during the larval stage, the temple.
Yes.
And so this muscle, the so-called myotomal muscle, which forms the tail of the muscle.
Yes. Yes.
And so initially they are kind of mononucleated. And as a matter of fact, we culture these cells very often. So when we take it out into the culture dish, they never become multiple nucleated. But in the body of the tadpole, as it grows, then if you use happy staining, then you find that it becomes multiple nucleated.
Yes.
And of course, this is because of the inclusion of the satellite cells, the
Yes.
yes. Right. And I'm just wondering, in zebrafish, do you have such a process? So in other words, these muscles you look at, you said that they kind of disappear. And whether it kind of goes into another state, I'm just fantasizing.
Very good.
Multi-nucleated,
yes.
somehow loses the presence or whatever.
These are all good questions.
Incorporating the satellite cells.
Yes. So, very good question. So there, due to time constraints, I cannot show you the full set of data. So actually, this for the actual paper, we did exclude the possibility that simply the fluorescence just disappear. So in the end, we found that there is a specific cell death mechanism involved in this process. It's not case dependent, but the autophagy mediated cell death. We see a very strong increase of ATG5 and ATG7 in degrading muscles. Maybe I should not use the disappear, they just eliminated. So we can catch the degrading, the eliminating process in real time. So we do checking these muscles from the intact form to the eliminated, catching the entire eliminated process. But it's not simply because of the cell lose a label.
Yeah, well, this is very interesting because if you look at the ignorance, the tail essentially undergoes metamorphosis. The tail is resolved.
Yes.
I mean, I guess it's destroyed.
Yes.
And so in the case of zebrafish, they don't go through this process.
Yes.
But there must be some kind of related metamorphic event. And this could be also due to thyroid hormone too, right?
Yes.
And so this process, maybe due to thyroid hormone, and then somehow it will be possible to eliminate it.
Very good question. So that review is answers exactly the question you just asked. So firstly,
I'm not a reviewer.
the first thing to point out this process are they similar to the tail, during the development, the tail will be long, and then later so that people also document the massive muscle replacement going on. Right? And then that in our case, we want to highlight because what has been documented in temple is only tail, right? And nobody knows about other part of muscles. But we are seeing this on the whole body scale. Doesn't matter if it's craniofacial muscle or swimming pectoral muscle or trunk muscles. The whole body elimination. So this is I think a very different. And also the second question also is good isn't this require some kind of trigger like hormone? Thyroid hormone has been shown in the dental side is the key for the trigger the metamorphosis. All these morphological changes because of thyroid hormone. And the changes we see in the fish, the stage we saw seems much earlier than the actual juvenile growth period. But we also will test, actually now we are currently testing that to see whether the hormone treatment can accelerate the process or not. But our initial, now current idea, we think that this probably is more of a early development, and autophagy related, maybe related to some physical property changes that we haven't connected with hormone yet, but now we are doing this. Thank you very much.
Thank you, that was amazing. So I have two questions. The first question is, I keep thinking about the chromosomal segregation defects in your skin cells. I think they all have, I know that you said that they all have like characteristics of like abnormal cell division in cancer cells, and another characteristic of cancer cells is that they have aneuploidy. So I wonder whether aneuploidy could have played a role in such abnormality in what you observed. And my second question is about the muscle. So what's the difference between the larval muscle and the adult muscle, so that it's beneficial for the fish to like replace all the myofibers when they grow?
Very good question. So maybe I will go to the second one first. So this slide, we also are doing the revision. This is also experimentally we focus on, because there's such large scale replacement going on. And why bother the animal to do that? Why not to keep using the pre-existing muscles? Because muscle growth, before people know about such large scale degradation, there are two muscle growth mechanisms that you can see on this group. One is hyperplasia, one is hypertrophy. Hyperplasia means the satellite cell, right? We always, during this growth period, continue to, from satellite cell, undergo a symmetry division to make new myofibers. The other one is satellite cell will fuse with the pre-existing myofiber to make the muscle bigger. This is also considered part of the muscle growth mechanism. But we found here, right, since there is a very drastic event going on, right, they just get rid of all the larva, right, and grow everything anew. And I want to highlight, right, actually, if you think about the metaphor I talked about the human teeth, you can see, right, human baby teeth and adult teeth. Why do we need to have such replacement going on? Because teeth itself, right, is a highly differentiated specified structures. The baby has certain diets, right? So the baby teeth are good for them. And as an adult, we need a totally different diet. Right? We need adult teeth to serve the specific function. In order for this to replace, to grow such a differentiated structures, right? Instead of a remodel the existing one, why not to make everything new? Right? That's the idea. So to address that, right, actually now we already have done the C4 assays, trying to measure the metabolic changes, right, on the larva and the adult myofiber. And we can see that, right, there is a very distinct difference between these two populations. So this is why there is such replacement going on, because we need a stronger muscles. We need a more metabolized, more active muscles at adult stage. And instead of remodel the older one, and then do everything anew in the And go back to your first question. It's about aneuploidy. I think aneuploidy, this term also is being used, I think, quite loosely. When we say aneuploidy, simply just means the cell has a normal chromosome numbers, right? And in this case, right, they somehow go into another extreme. Right? It's not just a normal missing one or two chromosomes. It's missing half and even more, right, in the second division. Yes, so when we decide to describe this, right, in our writing, right, we call it hypoploidy. We don't call it aneuploidy, right, for a reason.
Before they start a synthetic vision, like are they already aneuploidy? Because that's why you're going to see all those like spindle defects and like
I see. So from our line to data, right, we don't know whether the division we capture, right, is the first division or the second division. So that we, I really have no idea. Thank you.
Thank you for the talk. I would like to ask that when the skin cell undergo a synthetic fission, how do other tissues keep up with the right connection with the skin cell? Would they feel like separately, or because one of the skin cells like growing really fast, and what happened to the other cells? Will they keep up with them or like doing separate?
Okay, yes. So here, let me explain a bit about, let me rephrase your question. So your question is when this superficial epithelial cell undergo a synthetic division, what's the other surrounding cells, right? Or how do they respond to such division, right? And then here I need to explain further, right, about the structure of the fish skin. So fish skin, right, just like our skin, is stratified by the structures. And so what we found in especially the developmental stage we look at, right, at this stage, the fish skin is about 2 to 3 layers. So basically, like when this cell divide, actually the cell beneath them, right, oftentimes they are stem cells, basal epithelial stem cell. So actually they undergo the normal cell division. So you can see that, like, think about you have a stratified structure and try to expand rapidly in 4 dimensions, in all directions. The bottom one already work very hard, undergo typical cell divisions. And the top one, right, in order to maintain bi-layer stratified structure, the bottom one somehow being forced, the top one is sensitive, somehow being forced to do a surgical division. And after day 14, right, skin will transition, undergo a transition from 2 layers to 3 and 4 layers. And all of a sudden, such division will be become much less prevalent. So we think that undergo such divisions, right, maybe also facilitate the skin cell shaping, and then later, right, that might also regulate the following stratification at a later stage. But these are all some of our speculations. Yeah. Please.
Thank you for your talk. I really like the input approach. So I have 2 questions. So let's say we have one cell and it went through an synthetic division to give 4 cells.
Yeah.
So we said that the genetic material would be divided into these 4 cells.
Yes.
Maybe it's not equally, but are these cells are still functional in terms of performing some functions? Because we imagine that the genetic material is different maybe in the 4 cells. So are they still able to function somehow?
Yes.
Yeah.
Okay. So this is a previous question. So basically, we now already have preliminary single-cell sequence data. We found that indeed the DNA, the chromosome number is further reduced upon each division, and the segregation itself appears to be total random. So there is not chromosome 1, 2, 3 go to the other side, chromosome 2, 3 go to one side, no random. Right? And then now when you try to define a cell function based on genomic content, right, we need to be very careful about this, because like red blood cells, right, it has absolutely no DNA content, remain very important and remain functional for 120 days in vivo. Right? So to this cell serve their specific purpose, they say, of course in our case, we speculate it's important, these cells are important to maintain the barrier function. This is, but now we haven't had the direct experimental data to support that.
I was thinking, is it possible to do RNA sequencing after these divisions to see if we have a new synthesis of some important proteins?
Yes. So now, we did do the RNA sequencing, right? The cell sorting, we do the RNA sequencing on cells like that have no at the face, right? No synthetic division at all, versus the face, then we see a lot of synthetic division. And of course, like any RNA profiling experiments, we see a lot of genes go up and down. But we do notice a few candidates, that apparently they are part of the cell cycle components. So now we are testing these specific candidates in our system. and to see like whether they can by overexpressing these specific genes, right, can we promote aesthetic vision or not? So they might be able to support like maybe this is whether this is an active regulated process or passive.
Thank you. Thank you.
Thank you for the talk. So I'm wondering, do you have any hypothesis about how do these skin cells escape the cell cycle checkpoint at such a specific developmental stage? So is it like the cancer cell, or like, as you mentioned, the tension is an important factor, so is that a possible stimulus for such phenomenon?
Yes. So these are also very important questions, because we got asked to analyze that, because now it's almost like a dogma. The cell division must be tightly controlled in vivo. And this type of cell division, actually people have seen this, right, in vitro, something alike in vitro already. So we found that there are some more literature. People put a lot of cell cycle blockers and some cell checkpoint inhibitors in cancer cells. But at the same time also give them high nutrition to push them to proliferate. They do, sometimes people do report cell can divide without the DNA replication, but that's all in vitro scenario. And why this would happen in vivo, right? Really, this is a very an outstanding question that we need to answer in the next phase. You're right, possibly like Eugene talked about, like could be the tension is extremely high on these cells. Can we test this with some in vitro models? Or during the skin stratification in the mouse model, people show that in this process, right, the skin cells on different layers, right, actually they will have different physical properties. The phase separation, or the pH value changes, because somehow like this cell, when they close to the top, right, they become very acidic. Right? It's all these some other another layer of the regulation to make such division possible. And somehow when those scenarios happen, and then they can somehow shut off the cell cycle checkpoint with some kinase regulator. Right? These are all very exciting direction, I think, to comment.
Thank you.
I'm not a cell biologist, so I asked a very naive question. Are they still doing translation?
Translate.
If they are doing translation, suppose after a couple of days they will look very different, right?
Yes. So this question also is interesting, because now we found that from the meeting, right, there's a bottleneck for other people to study this type of division, because such a sophisticated genetic tool, right, difficult to implement in a different model system. And from our imaging study, right, because we track this cell for long times, we track the one single cell for 3 weeks, and sometimes even for months, and we noticed that these cells, right, before and after divisions, right, the fluorescence labeling is always very stable. Right? The green cell will continue green, the purple cell will continue to be purple for like 3 or 4 weeks period. Right? So that leads us, right, to speculate, right, there may be the protein, right, for this cell, right, the degradation rate might be very low. And maybe there is also not much new protein being synthesized, because they already have no DNA content. But how could their fluorescent protein color maintain? Right? If they make new proteins, right, then their color will get changed. Right? So we now are trying to build a new reporter cassette, right? Trying to see whether we can create another system to monitor such divisions. For instance, one of the cassettes we look at, right, is the KKGR. The KKGR is photoconvertible protein. Usually people use it to do the transient labeling on cells. Usually you convert any cells because it's the photocomputable mechanism, right? Oh, let me slow down a little bit. So then the reporter construct called KHDR. And you people use it, right, to monitor, to do the cell behavior tracking. And the mechanism, right, is the protein itself, right, would appear in green color, but if you shine a specific UV light on the cell, actually the fluorescent protein will turn from green to red, permanently. And then after that, any new protein being generated will continue to be green, but the pre-existing one will be the red color. And when you use this type of construct to label any cells, actually you will see, right, every time after you do the conversion, the cell will, all of a sudden after conversion, turning from the green to red immediately. And then later, right, they will turn from red to yellow to green in 2 to 3 days period. Of course, different cell types is always like that. And now we want to test, right, with this reporter, during our settings, right, is it possible after conversion to become permanent? And then we want to use it as a direct readout, right, to comment on whether the protein degradation and protein synthesis, right, somehow it become a compromise during this cell division.
You have planned to make a calm heart or a calm brain.
Yes, very good. So we the palm heart actually similar idea, right? Not similar system, but similar tool, right? It's being used actually by another group actually by former mentors. They already use the older version of the rainbow, right? Already to use it to study the heart development. Right? So they found that in heart development, right, they can identify the dominant cause, right, during the post-embryonic growth period. Yeah, and then that, like, of course now our system now, like, the resolution will be even higher, but I'm not so sure we will go back to it or not. Because another thing about heart, right, it's difficult to study under the blind imaging settings. And now we somehow focus on doing everything in lab. But that, so that on the heart part, like, already has been done. And now the lab, we do our building tools and try to look at the neuron cells. We try to see whether the neuron cell, people always assume at certain stage there will be no new neuron growth or proliferation. If not, we see that in total. Can we see some of them actually are proliferating in some of the places? Because now we are seeing a whole fish.
You may convert Professor John's lab into a neural fish lab, right?
Totally.
So I have a question. How does this stem cell know I should divide once or divide twice? And are they all classed together or randomly distributed?
Yeah, okay, good. So in the study, we did look into that. Right, so you can see, right, sometimes the data I show here, right, is a bit over simple. Right? If you look at the fish growth, the fish, has an anterior part, has a posterior part. They have chest, like us, they have chest, and they have the posterior tail part. Right? Their chest part, usually the bottom is bigger, and their tail part, like, is thinner. Right? So when we quantify such division along the anterior posterior axis, we see more division on the anterior part when the body surface is bigger. And we do see less division on the posterior part when the body surface, right, is less. So since with the whole model, the whole field, right, somehow this division provide a very frugal yet dynamic regulatory mechanism. So if you need more surface, or just simply divide more, then I have more surface. I can have more equal surface to cover body, to the body, certain body regions. And if I don't have a lot of body surface to cover, then I don't have to undergo such division. So we can see later body region specific regulation on such division.
Before I end this section, I just want to remind everyone， 提醒大家2件事。第一件事情，今天下午1点半的时候，在这个丙前讲堂有这个陈老师要分享他的学识历程，好，欢迎大家一起参加。然后有参加的同学，你可能都有机会得到就是星巴克的券。然后第2件事情是我们等一下在旁边有午餐，然后